The issue of chemicals in the environment is a story about science, politics, and humanity's relationship with the planet. My name is Chris Hughes, independent scientist, and this is the Chemical Journeys podcast. Join me as we uncover the story of chemicals in the environment through conversations with individuals working in this space. So, let's get started. Hello, and welcome to the Chemical Journeys podcast. My name's Chris Hughes, and I'm here today with Delina Lyon, Science Executive for Environment from Concawe. Del, thanks for joining me.
Thank you, Chris. It's a pleasure to be here with you.
I'm really excited to have you on the podcast, Del, because as you know, we've known each other for quite a long time now, having worked together first as colleagues at Shell and then various interactions since then, since I've been working as a consultant. One thing I can say is that it's always been really good fun talking with you and working with you. And so I'm excited about the topics that we're going to cover today.
Excellent, me too.
So let's kick off. I think it'd be helpful for you to just explain to the people listening who probably haven't heard, what is Concawe?
So Concawe is the technical and scientific division of the European Fuel Manufacturers Association. So this is an association of refining and renewable fuel manufacturing companies. So it's been expanding its scope, right? Because now that we have the new feedstocks coming in, renewable feedstocks, we are also trying to include those. But Concawe itself was established way back in 1963 by oil companies who were trying to understand the environmental impact of their activities. So over time, of course, the scope of Concawe's activities has gradually expanded with different societal concerns over health, environment, and safety issues. So now what Concawe does is do scientific research and other types of technical studies to cover different topics related to the refining industry. So the transition toward carbon neutrality, for example, safety, at sites, air quality, emissions of waste to air, to water, occupational health, all the different types of fuels. So that would be for road transport, for aviation, maritime, and then also looking at pipelines. But there's also another part where Concawe works on the regulatory mandated chemical registrations of the products that come out of a refinery. So I thought it would be helpful if I give like a quick description, the refinery 101 in about 2 sentences. So you have the crude coming in. So this could be a petroleum feedstock. It could be one of the new renewable feedstocks. But what's coming in is a lot of hydrocarbon molecules that span a huge range, right, of carbon number. And in the refinery, you're separating it. So if I simplify very much, you're separating it into different fractions. And I say I'm simplifying here because you can have other manufacturing processes. But what you're trying to do is refine the carbon number range of each of the fractions. So if I think of it with my EcoTalk hat on, I'm thinking about what's the carbon number range of each of these different fractions. And so each of these different fractions will become a different product, like a fuel or a lubricant or waxes. But in Europe, all of these products need to be evaluated for their health effects, their environmental effects, and to be able to calculate the risk that that product poses. And then this all has to go into a REACH registration. So REACH is the chemicals regulation in Europe. And so my role at Concawe is to help coordinate the environmental sections of what is termed the registration dossier. So the assembly of all of this information that you need to understand environmental impacts of the different products.
No, that's great. And obviously the discussion around climate change and fossil fuels is something that everybody will be familiar with. But I think what's really interesting about the work that you're doing within Concawe is that we're not really focusing on that. We're thinking more about the health and environmental potential hazards and risks of the materials that are being produced and the regulatory implications and compliance challenges around that. And there are some really interesting and transferable things that Concawe is doing that are necessary and applicable across the chemical sector. Hopefully, we can dive into that a bit more in our conversation. But before we do, could you just flesh out a bit more about your role within Concawe? Maybe tell us what a typical
Yeah,
day might look like.
sure. So my role, I guess the name of my role is a science executive. It's a vague term, right? Like, what does a science executive do? Do I execute science? Um, yes. So as I said, so if I have an overarching umbrella of REACH registration, one might think that all we do is fill out requirements under REACH, which include things like toxicity testing with fish, invertebrates, algae, maybe microorganisms, doing some testing for biodegradation. So there are all these tests that are required. Seems fairly straightforward, but it really isn't, because the way that you need to run those tests, or how are you going to provide a piece of information, really comes down to understanding the substances, understanding the tests, and then trying to figure out the best way to run them to get the piece of information that you need to show, yes, this chemical is or is not degradable, or it is or is not toxic. This is particularly challenging for fuels, for hydrocarbon fuels, because you're not dealing with one chemical, you're dealing with ranging from hundreds of different chemicals mixed together to millions or billions. Who knows how many when you get to some of the really complex things like a bitumen or uh, a lubricating oil where you've just got a lot of things in there. And this is because of the way that the product is made, right? So, number one, you're coming from something that's very complex. So, crude oil, right? There's all sorts of stuff in there. And then you're trying to refine it. You're trying to take fractions of it. You might have some reactions to clean it up or to optimize one type of chemistry or another, but you're still dealing with something that's quite complex. And so when it comes time to saying, okay, here's this mishmash of chemicals, is it biodegradable? It's not a straightforward answer, right? You'll have to say, well, maybe this constituent is, maybe this other constituent is not. But how are you going to tease that apart using one test or, a limited number of tests so that you're not testing or running thousands and thousands of tests? Trying to figure out those kinds of problems is a big part of our day, and then trying to run the tests, and then also coordinating with the different member companies of Concawe to get their input to see what their issues are so that we can start working on those. And then there's also a part where we interface with the regulatory environment. So we participate in groups where we can provide feedback or comments to the regulators as they're developing guidelines or as they're trying to implement changes. And then there's also an interface with the scientific community, which I'd like to think we are actually part of. But you typically think of it as like industry, academia, and regulators and NGOs. So trying to understand what everybody else is worried about so that we can anticipate as much as possible. Unfortunately, it's hard to anticipate everything, so I often feel like we're playing catch up or putting out fires or trying to figure out like, oh, is this really a problem or not? But it's a busy day, but I get to do a lot of things, and I don't get to be as much of a deep expert in any one thing. I have a lot of surface knowledge or awareness of multiple things, but then it's also trying to say, okay, who can we work with a little bit better? Who do we need to talk with more on specific topics? So there's a lot of like prioritization of what we need to work on because we are not a big group. From the environmental side, there's three of us, um, and covering a lot. So
Yeah, having spoke with you in the past, I'm aware of how busy you can get trying to keep on top of all these different activities that you're responsible for in that role. So well done for doing that. And I wouldn't do yourself down in terms of your ability to play a role as an expert in some of these topics as well, because you've certainly brought a lot of your own expertise to that role within Concawe. Just so that we understand it from a property standpoint, so you're looking after the products that are produced by the refining sector, and you're looking to understand the environmental hazards and risks. So from an environmental point of view, it's ecotoxicology and
Mhm.
to different organisms in the environment and environmental fate. So where do these chemicals go if they're released into the environment, and how do they behave? Anything else?
So we do also try to act as a resource for the other groups. So, for example, we work very closely with the human health group. So, if they have questions regarding things that we have expertise on, we can help them out. If there's for example, with the water group, if they have some questions regarding the testing, we can help out there. We
And the
also
water
try
group
to,
covers something different.
yes, so there are other regulations, other environmental regulations that are related to water quality. There's, of course, a relationship with what chemicals you're bringing in to what your water quality is like at the end. But we do sit as two separate groups because we deal with different regulations.
Yeah, and it's kind of like two lenses, right, to all this environmental management, chemical management sort of space that we're working in, right? There's the, I guess, the prospective end, which is the end that you're kind of sitting on, which is looking at, what are we making, how are we using it, what are its properties, and therefore, what impact can we foresee? And then I guess at the other end is more kind of like a retrospective lens, which is, looking out into the environment, looking for what chemicals we're finding, looking for what indicators of effects we might see, and trying to detect whether harm is being caused, and then working back from that to where that harm might be arising from. Is that kind of
Yeah,
a good way to kind of split the two roles as well?
yeah, I think that's really fair.
Hmm. Yeah, and you mentioned as well about your links with the human health. I mean, are you finding that those links are becoming more and more important as we go on, and we're talking more about things like the exposome and One Health initiatives? Looking at everything in a more holistic way.
Yeah, yeah, definitely. Also, because there are new hazards that are coming to the forefront as being of interest. Of course, for example, like with endocrine disruption, that's a new one. It's not a new hazard, but its incorporation into some of the regulations is relatively new. And there we're dealing with both human health and environment, possibility for endocrine disruption in both, but there's a clear relationship between the two. And so, if you can show that there's endocrine disruption, for example, in a mammal, there might be the same pathway in something that's relevant for the environment and vice versa. So, we do try to coordinate more. I have to say, historically, the focus has been on human health for toxicology and the
Mm.
interest for chemicals. Lots of, like, is this bad for humans, etc. The focus over the years, I think, has shifted a bit more towards trying to understand what happens in the environment as well. There's a big component of man via the environment,
Mm.
human exposure because of what you're having from an environmental versus I think where things started off with more with a occupational exposure or industrial hygiene aspect where you're worried about what happens when you are using a particular chemical. And so there was lots of interest there. We do make jokes sometimes about how the human health group just has to deal with humans and the environmental group deals with everything else. I think, still there is a lot of interaction that has to happen there in order to get the best science because they've done so much that we can leverage.
Mm.
It would
Yeah,
just be a shame to continue in silos.
definitely. Yeah, I think more than ever, it's imperative that the different groups talk to each other, and I can definitely recognize some of the rivalries and the teasing that goes on between the different disciplines. We as environmental scientists, we're often lumbered with the task of not only assessing hazards but assessing exposure as well, and being masters of environmental risk assessment tools and models, whereas on the human health side they often have occupational hygienists to do that for them,
Right.
right? So
Yeah,
but
and
yeah,
it's a
no.
real shame about the exposure aspect of things. I have to say, like, when we are talking to ecotoxicologists, we have a good amount of understanding, a good amount of people who are ecotoxicologists. They understand the hazards and stuff. When it comes to exposure science, it's so complex. And it seems so fluffy to people, right? That I think it's a little intimidating to say, "Oh, I'm going to understand the exposure, of all these chemicals in all of these compartments." And we do, I think, suffer from a dearth of expertise from exposure science.
Mm,
And I think we might see that it leads to then, more focus on hazard because
mm.
that's what people are comfortable with. But the exposure aspect is a huge component and it's
Yeah, and
difficult.
it seems like a trend that's unfortunately going the wrong way as well. I think if we compare to the amount of, well, at least on the human health side, the amount of expertise that was around sort of 10 years ago and the amount of effort that went into implementing exposure aspects under the REACH regulation, it seems that information is not always being followed up. And as a result, the discipline itself seems to be a little bit in decline from what I'm hearing. So thanks very much for that overview. I think it really helps to give a flavour of the nature of the work that you're doing, and we will dive into some of these topics in a bit more detail. I think it would just be great to understand a little bit more about your background and the journey that's brought you to where you are today. Perhaps you could tell us a little bit about that.
Sure. So my background is actually I started off with microbiology, and it will ever remain my first love. I was kind of the reason I was drawn to microbiology was, way back when there was the probably dating myself now, the Exxon Valdez oil spill. And I remember reading about it or hearing about it when I was a kid of indeterminate age that they were looking at using microbes, bacteria, to help clean up the oil spill. And I thought, wow, that's really cool. That's what I want to do when I grow up. My dad he did work for Esso at the time. And I remember when I was growing up thinking, I am not working for an oil and gas company, and I am not going to become an engineer. So, go to school, go and I got a master's in microbiology, and where I did research on, basically what are some of the genes that are encoding for degradation of aromatic compounds. And while I was going through the program, I sort of felt, I want something just a little bit more, I don't know, more applied, something, but I didn't know what it was, and I didn't know how to get there. So I ended up with a master's. I went and worked for a couple of years in a laboratory at Rice University, and there it was in the environmental engineering department. And I saw that they were doing things that were a little bit more applied. I thought, okay, this is what I want to be. I want to be an environmental engineer. So then I started my PhD program there. I ended up working on a project on nanotechnology because it was a new topic at the time. So I did like some very beginning work on the toxicology of nanomaterials, focusing on fullerenes, right? So it's a lot of fun. Um, and that field took off. This was at the beginning of that era of like, oh, what happens when you have nanomaterials in the environment? But I didn't want to stay with nanomaterials. I wanted to try other things. So then I went and did a postdoc for a couple of years at a lab in France at the École Centrale de Lyon. Yes, Lyon, and yes, my last name is Lyon. It was very confusing for everybody. But I was there for a couple of years in a laboratory that worked on microbial genomics. It was a really fun lab because half the lab was an electrical engineering lab, and the other half was this microbiology lab. The electrical engineering folks were studying things like what happens when you have lightning strike an airplane. They had this big lightning generator in the lab, which we then thought, if we're in the same lab, could you please shock my plate of soil that has some DNA on it and we can see if it goes into the bacteria. So we did that kind of stuff. It's really fun. Um,
Trying to create life.
it was exactly like what happened at the beginning of time. But anyhow, so then did a postdoc for a couple of years, went back to the US because my husband had a job with National Cancer Institute. He had a fellowship there, and I went back pregnant. And then this was kind of like the big the big change in your life, right? So when my son was born, he had special needs. We needed to figure out what were we going to do when we grew up and all of that jazz and try to make some decisions to have a more stable and get him some good services and stuff like that. So I started looking for another job. I had been working at Howard University, which is in Washington, D.C., doing again some more work on nanomaterials. But that's when I interviewed with Shell and then ended up working at Shell as an ecotoxicologist. But I came in obviously with, microbiology background, more on biodegradation, bioremediation, less so on the actual ecotox aspect. So all of that I have tried to pick up along the way of being an ecotoxicologist. And it's also kind of interesting because coming into the field of ecotoxicology and particularly regulatory ecotoxicology, where you're saying, okay, is this chemical bad? How are we going to find out? Let's see if it's going to kill a bunch of stuff. And then you have your dose response. You put in this amount of chemical and you see how many of the organisms die. Coming from academia and sort of thinking about the state of science and how much information we have and how much we can understand like mechanisms of why is something happening on a genetic level, that richness seemed to me very distilled, right? All of a sudden it's the,
Mm.
I think they call it the kill 'em and count 'em approach. And so it was like a little shift of a mindset of going from, oh, you could understand this complex mechanism or this gene interaction or something like that. But when it comes to trying to make a decision, are we going to use this chemical? We're going to expose millions of people to this chemical. How are you going to make that decision? It's really hard to do it when you're trying to use information that is vague or subject to interpretation. And
Mm.
so that kind of kill them and count them, very straightforward, right? You can see a number, you can compare it across chemicals, and then you can make a decision, do we use this chemical or not? It was a shift in perspective and also priorities of trying to understand what is really important and how can this help make a decision or support a decision in the end. So
Yeah.
yeah, so then I ended up at Shell. While I was at Shell, I was seconded to Concawe for a few years, and then I ended up leaving Shell and staying at Concawe. Yeah, that was pretty probably more
That's
than you
great.
wanted to know, Chris.
No, no, it's great. It's great. And yeah, you, it was really good. I remember when you joined Shell, it was great to work with you on the EU reach aspects because you came in really early on, working across the pond a lot of the time with us on
Yeah,
those things. And I think, and I'll attest that when you came to Concawe, you definitely had an impact in terms of your ambition to drive the research forward and address questions that were kind of barriers to making progress on some of the regulatory challenges. So, you've had a really good impact. So I'm glad that in a way you came back to the oil industry armed with the
Right,
tools
became
and
an
the
engineer
knowledge.
for the oil industry, right?
Yeah, to hopefully make a positive impact. You mentioned there about the shift from academia to the regulatory science environment. And I think that you're definitely right in seeing that contrast between what we can do in academia in terms of the questions that we can investigate and go after and the insights that we can gain versus the need for tools that can help to make practical, robust decisions about how we manage and deal with chemicals. What was it like making that shift from academia to industry? I mean, did you find it frustrating? Do you have any kind of key reflections? Do you think overall it works well? And how have things changed over time?
So when I had started out, I was based in the US, and so I was really more involved with TSCA. So the US chemical regulation, the Toxic
Mm,
Substance Control Act. And while I was there, it underwent a big revision. And so I could see what kind of some of the changes that were being made and what were the justifications for those, and how sort of this process of trying to make a practical implementation of science. Coming from academia, you want to understand something to the nth degree, right? When you're trying to explain what's going on, you want to have hypotheses about, like, so here's what's going on here. How do I demonstrate that? There's this drive to understand 100%. But then trying to come in and say, okay, we have to register this chemical, we have to fulfill these particular tests. Every time you run a biological test, something happens. Like there's
mm,
always something that goes on that is difficult to explain or you don't understand exactly why it happens. And the academic in me would want to go back and, oh, well, let's just repeat it. And let's, do we get that again? Or what is it that's driving that? But for a practical standpoint, does it really matter if you're trying to demonstrate, uh, the toxicity of something? Does it matter that, you had this variation in your control? So trying to understand, like, okay, at some point, you have to make a decision. And I think while that is hard for us, the folks who are trying to make registrations, I think it is 50-fold harder for the regulators who are receiving this information
mm.
because we provide it, but then they actually have to make a decision, and
Yeah. And
and
they're
they
bound
don't have the
by the
ability to.
precautionary principle to make conservative decisions. And so often there's a source of disagreement between
Mhm.
different groups about how data should be interpreted or what data can be used to support a certain conclusion versus not.
Yeah. So I think even though there's oftentimes a lot of complaining, right, of like, oh, this is too strict, or oh, this is that, there is an explanation. I think everybody is trying to make the best decision that they can given the information that they have and the limited amount of time and resource that they have to make that decision. So I think that
Yeah,
might be the difference for me from academia to something that's super applied and trying to be very practical is you just have to let some things go.
yes, yeah. And I think as well, with the academic side, the sort of scientist burning within all of us, we can often become optimistic about the data that we're seeing or how it can be interpreted, and then that could be a rude awakening when it's presented and a regulator might take a different view. I could certainly say I've been in that situation before where I've looked at a set of data and I thought this is pretty clear for me, and
Hmm.
then comes back and somebody else has taken a very different view. I think people might not appreciate that within these assessments, whilst they are intended to be rational, objective assessments, there is by necessity degrees of subjectivity to them. And I think there we enter into the domain of what's called weight of evidence, which, is a really important term in our field. But yeah, I think it doesn't always get used in the way that it's perhaps intended.
Yeah. And I think also when you talk about subjectivity, there is always a human element to all of these decisions. There's also a human running all these tests. And so, where it would be very nice to be able to make decisions in a consistent, clear, fair way without any kind of bias, that's impossible until we train whatever AI machine to do it. But even then, there's going to be somebody who's behind that training of that AI machine, right? So um, the human component to science, I often think is very interesting because think about the zeitgeist of disbelieving science. I think that's the pendulum swimming the other way because of some of the realizations that, oh yeah, maybe science is not 100% and there's a lot of interpretation, there's a lot of human error that goes into it. Yeah, I think this is going far afield of where we started though. But
No, that's fine. That's what the podcast is for, to have meandering conversations. I'm sure people find it insightful. I guess another point is around how the research is translating into regulatory processes, which we're sort of dancing around that topic anyway. But, we talked about an example of where some data is being generated and you think it's going to become useful in regulatory context. And then this question around, or this objection around the relevance of that information to make a regulatory decision, prevents the data ultimately from being used. And there's degrees of freedom there in terms of the discretion of the people who are looking at the data in terms of how they judge that. And it's not always super clear or transparent as to why certain judgments have been made. But similarly, on the other side, there's a huge amount of science being done looking into effects of chemicals, and there's a huge amount of new research being generated and new insights. And some of those insights could point to new concerns around chemicals, new effects like endocrine effects. How is that translating into evolving regulatory policy, would you say? Do you have any insights on that? And I'm thinking in terms of the pace of change, and also a shift in the culture around science, in that, scientists are being pushed to advocate more in the science policy interface. And we have social media now, which is changing the way that we're all communicating and receiving information too.
mhm. Yeah, so I guess if we're going to make a little map in our heads, it would be, the idea is like, oh, the academics come up with the science, and then the science gets translated over to the regulator to come up with, like, here's how we're going to run the tests. And then that is imposed upon the industry to run the tests and generate the data. But it doesn't work out in that kind of neat, right? It ends up sometimes it's the science that the regulators are interested in. They have a question, they go out and they look for the answer. Or maybe it's industry who has a question, they go out and look for the answer. And I think there's a frustration from the part of academics of like, well, why aren't you using this piece of research that I have
Mm.
developed that could answer your question? And it's that translation from an academic piece of work into something that can be used to make a decision, right? And something that can be used to make a decision for lots of chemicals. So I think that's where some of that frustration back and forth between the people who need the science and the people who are developing the science arises, is because
Mm.
a lot of things work well for a few things, right? So it's this idea
Yeah.
of the domain of applicability of your test or of your approach. Will it work for a large number of chemicals, or is it just going to work for like one or a few that have a certain structure or a certain moiety that allows that test to work?
Hmm. Hmm.
Whereas the regulator needs something that they can just apply to lots of chemicals. And I think that's where things get difficult. Then when you, as a scientist, might be asked, oh, well, I want something that's going to work for tons of things, you have to compromise a little bit on how well that test works then. Because if
Mhm.
you want to be able to accommodate lots of different things, maybe the interpretation gets a little fuzzy for certain classes versus others. So, yeah, I guess I understand why there's a frustration back and forth.
So, just trying to explore and reflect with you on how science is evolving and translating into regulatory practices moving forward. And you
Hmm.
mentioned there
Okay.
that, the frustration between academia feeling a bit left out of the overall kind of process. And I remember reading papers from a few years ago where that was being expressed quite strongly as to why scientific research isn't being incorporated. And I think there's been a big amount of progress made since then in terms of helping to translate academic research into something that can be used for regulatory purposes. And part of that has been a big part of that seems to have been tools for evaluating the quality of the information in terms of its reliability and its relevance, and encouraging academia to perhaps raise the standards of its execution and its reporting of research so that enough granularity in the data that's being generated
Mhm.
and the metadata around it can be recorded and assessed by regulators. And I know of our own initiative in that space with the persistence assessment tool, which perhaps we'll come on to. But I also sort of reflecting that it seems that ECHA has made quite a lot of noise recently about, sorry, and ECHA is the European Chemicals Agency, noise about the need to build dialogue and improve collaboration and hosting workshops, for example, most recently with the contract research organizations across Europe, which I think that and also issuing its research needs report, which is giving a clear signal to researchers of what they think the gaps are in regulatory assessment. So that overall to me seems positive. And there are also issues of trust ultimately, aren't there, between different groups, which I think probably a couple of years ago bubbled over. I don't know whether they're still bubbling over or whether they're simmering down a bit. It feels, mate, I'm quietly optimistic that the temperature might come down a little bit.
Yeah, so I guess we would also be remiss if we didn't mention the OECD, right? So, in terms of like, there's not just the regulators, there's also the United Nations, the Organization for Economic Cooperation and Development, or they have a suite of testing protocols that are supposed to be globally accepted, right? So it allows chemicals to be tested in one area using these standardized protocols and then applied everywhere. These protocols, therefore, have a lot of power, importance, I guess is the word I'm looking for.
Yes.
These protocols are important
They're
because
internationally recognized.
they're, yes, and how to get the academic research in a shape that it could be developed into one of these OECD protocols um, can take a very long time. Right? So I think you and I are familiar with one that has taken over 10 years and is still not finalized, whereas there are some that can go very quickly. And
Mm,
so it's a question of, I think, who is pushing for them, the interest of having those protocols quickly and readily available. I think one of the ones that has gone relatively quickly has been the one for bioaccumulation in Hyalella, the HYBIT test.
mm,
And
mhm,
there was an interest because the HYBIT offered a non-animal approach for bioaccumulation testing. And the push towards replacement of animal testing has been like huge. It's
mm.
been something that people have wanted to do for a long time, but I think also it has proven to be difficult, both because there have to be tests available, but there has to be this confidence in the testing. And
Yeah.
I think that's the same for any kind of test, whether it's a non animal approach or a new alternative method or just one of the old tests. But there's this idea that old is good, I guess. We're
Yeah.
used to the old version. So, but I think the question you were driving at earlier, I had just wanted to make sure that we mentioned OECD,
No, it's
but
very
I think
important.
the question you were driving at earlier was who and why should different groups be, and how, I guess, should different groups be involved in chemicals regulation, right? Is this
Mhm.
something where regulators and academic scientists and NGOs should be developing the work in the science, and because industry has um, an, a conflict of interest, some would say, but do stand to benefit economically, should not be allowed to participate in a discussion that's going to affect them, right? So that's that whole idea of conflict of interest. At the same time, those who know those chemistries that are being regulated are the people who sit in industry. Those are the people who have been doing lots of research on it and they understand how the system's going to work. Clearly, I have an opinion here, so
Mhm.
take it for what you will. But my opinion is that you would want the people who understand the chemistries and who understand, okay, if I have to run this test, this is how it's going to turn out, and this test is or is not going to work well for my substance. You would want those people at the table. How much influence do you want to allot them? I think is a different question.
Mhm.
But that their voices and opinions should be heard, I think is crucial because you can waste a lot of time and a lot of effort creating a test or creating a system that then doesn't work
Mhm.
when people actually have to run it. So,
Yeah, I think this is such a hot topic and also a very deep and complex and sensitive topic. I totally see where you're coming from. I had a very interesting conversation with Peter Fisk as well, which hopefully will be coming out, if it hasn't already, on this podcast. Something that came through is that, as scientists, if we're all within the profession of science, and it's often a vocation rather than a profession, then a core principle of being a scientist is to be objective, or to try as much as possible to be objective. That is a moral principle, a high moral principle that we have to uphold. And in doing so, we should also give benefit of the doubt to other scientists that we interact with, and judge them judge them in the same way. And in doing so, then in principle, it wouldn't matter whether the scientist was from academia or industry or the regulatory community. Now, there is the corrupting influence of interests, broader interests, which affect everybody. But it's not only about money. To sort of push back on the focus on financial incentives, and that there are other ways that people can accrue value from what they're doing, in terms of status, and we live in an attention economy with all this that's happening. So there are secondary ways that people can leverage wealth from the positions that they take, that's not through being paid directly by an employee relationship. So there are lots of, I mean, yeah, this debate is going to continue on, but I'm encouraged by, that there seem to be some attempts to bridge gaps. And I think things like scientific conferences that bring people together in person to have conversations are really crucial to that, because it's not good when people don't get to interact face to face about these things.
yeah, no, I think you raise a really good point, Chris, that I don't really know anybody who said, I'm going to be an environmental scientist because that is where the money is. Um, most people go into it because they're passionate about the environment. And that I get to meet a lot of people across all the different sectors. You do also. And I think the one thing you can see is that people are passionate about their work, they're passionate about the environment. And so when trying to say certain types of scientists should be excluded from the discussion versus others, I disagree. If we want to say certain, you know, you don't want to have your marketing expert at the table when you're discussing a scientific topic. That's appropriate. But yeah,
Yeah.
I agree with you that like there's a common thread. And you'll see that people, you can transfer sector to sector. Lots of people do it. They move from academia to regulatory to industry.
Hmm.
I haven't had as much of that kind of transfer back and forth, but it's the same person.
Yeah.
They
Yeah.
still
Yeah.
have the same knowledge and the same expertise as they're going back and forth. So why would one why would their speaking from one group be more reliable than when they're speaking for another?
Yeah. Exactly.
On a
And I think
strictly scientific topic, huh? Yeah.
of course, yeah. And I will say that you personally have done more than most, I think, to reach out and connect with people and build a network and to understand different perspectives as well. So I think we can veer back towards Concawe and the activities that are going on there. And we touched on it earlier on about petroleum substances and the challenges of assessing them due to their complex composition. And I think you may have also mentioned the challenging properties, although perhaps we can go on to that in more detail. And it's worth recognizing, I think, that whilst anybody who did science, I think at high school level, probably had the lesson on what is oil and how is it made up of many, many, many different constituents, and from
I
a
think your
hazard and
school was
risk
better than
assessment,
mine.
maybe the fact that from a hazard and risk assessment point of view, it's important to understand that composition because the composition will dictate ultimately the hazards and the risks collectively from the material that you're dealing with. But it's important to recognize that whilst oil and oil products are probably the archetypal complex material, they're not the only complex material that's used in commerce. In fact, recently it was suggested that between 20 and 40% of substances on the market in commerce have a complex composition, and I think that's before we start to talk about polymers, which are all
Hmm.
complex, have a complex composition. So the challenge of dealing with complex substances is not unique to petroleum substances, and a great deal of progress and work has been done to tackle that problem in the petroleum sector. So perhaps you can tell us a bit more about some of those challenges.
Yeah, so, and there's also a great group at HESI, the Health and Environmental Sciences Institute, that tries to bring people together to talk about these complex substances. So I'll go ahead and throw the acronym out there of the UVCBs, which
Yeah,
are substances of unknown composition, variable composition, biological origin, or complex reaction products. So they lots of different types of substances fall into this category. So yes, petroleum substances, some of the other fuel substances. You've got resins, you've got fragrances, you've got surfactants. What am I missing here?
natural complex substances.
Natural
Yeah.
complex substances. Thank you very much. And so we all share the issue of how do you do a test on something that has multiple constituents in it. The problem being, of course, uh, is it all of the constituents that are bad, or is it just some of them which are bad? And how can you make that decision if you're testing them all together? So there's kind of two schools of thought, right? You could
Mm.
be looking at the whole substance, or you could be trying to understand the composition and then look at the constituents separately and then kind of add everything up together. And you think of it as like a mixture. For something that is relatively simple, so maybe you have 3 to 4 different constituents, and maybe one of them is 90% and the rest are just a few percent each. You could look at them as different constituents and then add it up together or decide that this is the most important constituent that I want to focus on. For something like petroleum substances or some of the other fuels, that's almost impossible. because there are so many constituents. I think even from Concawe's portfolio, like the least complex substance still has hundreds of constituents in it. So doing the looking at the constituents one by one in the past has been almost impossible. We are trying to understand if that is possible. So, meaning that there are analytical methods that can give you a more simplified breakdown of the composition so that you can have maybe not constituents, but maybe groups of constituents. We have been working on uh, modeling that allows you then to say, okay, if this is this is the expected toxicity of this type of constituent, and if you have this list of constituents, each with this expected toxicity and each at this concentration, we have a way of modeling what the toxicity might be of the whole substance. So that's another approach. We've also been doing a really fun project where we are trying to run a biodegradation test with analytical that lets you separate out the constituents and then track those constituents over time and see if you can generate like a biodegradation half-life for an individual constituent. And is this approach, we have been successful in generating like thousands of data points in one test. So for us, that's fairly exciting. It does give people a little bit more insight of what's happening with your fuel versus trying to say, okay, well, as a whole substance, maybe we can say 90% of it goes away, but we're not sure what's what's remaining.
Hmm.
So getting a little bit more insight. And I think with the evolution of analytical methods, also with the computing abilities, because when you're trying to track lots of things or trying to even model, thousands of constituents, you need some computing power behind you to be able to do
Yeah.
that. And I think we're at that stage now where we're able to do a little bit more of a constituent level approach because of the analytical and because of the computational abilities.
Yeah, that's really the project you talk about, the biodegradation, where you've used those more detailed analytical techniques, is really interesting because that's been one of the debates that's been had about persistence assessment, isn't it? That you shouldn't test multiple constituents together because their degradation might influence each other, and so you should you should test them individually. But then you have this hugely impractical proposition that is, run a standard test for all these individual constituents, which is, completely unworkable from a practical standpoint. And a lot of the fear about whether they will degrade differently together is perhaps not well founded in evidence. There's a sort of an uncertainty there, and in any case, often that is a more environmentally relevant situation in that, these constituents exist together within a product and they're released together into the environment. So that's just one example of the complex and nebulous kind of discussions that can arise as we try and make decisions with the data that we have.
Yeah, and I think also with all of these biological tests, but particularly with biodegradation, where I think historically it's a little bit of a black box approach, right? You get an environmental sample, you put your test chemical in there, you then try to see how much of it has biodegraded. And the black box part of that is what are the microbes that are there? What's their concentration?
Yes.
What are their metabolic capacities? How much biodiversity are you capturing there? I mean, it's just a small one sample to make this big
Yeah.
decision over whether this is biodegradable chemical or not, which then implies, are we going to release it into the environment potentially? It's
Yeah.
a big decision to base on one small sample. Or
Yeah. Are we going to continue to permit this product, this material, to be used in commerce, big decisions are being made around these data. And yeah, another, I think you hinted at it there, a point that I think is important is, how well does a biodegradation test really represent what's going on in the environment? How does that degradation you're observing reflect what's happening in the real environment in a setting that the test is meant to represent? And you and I are both participating in a workshop coming in a couple of weeks, less than a couple of weeks, with ESETOC, looking at the OECD 309 test guideline, which is the surface water biodegradation simulation test, which, yeah, I think we have a lot to discuss about that guideline, right?
Yes, yes. And especially like, from so my background was in microbiology and sort of understanding microbial diversity in terms of biodegradation capacity and things like that. And it drives me crazy sometimes thinking about this. I think about it for too long. Like, why would you make this decision based on a few samples? You're not representing it at all, the diversity of environments or of microbial communities that exist. There's been, as you're well aware, lots of research showing that you even go to the same place in the summer and the winter, you're going to get different microbial communities. You take that sample out of your environmental setting, and you've changed the microbial community. All of this impacts the biodegradation capacity of that sample, right? So, yeah, but what is the alternative? Dun, 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 dun. I don't know.
Yeah, that's the upshot. And I think that's part of the problem, right? In that we don't have a good alternative. And I also think that overall progress in this area has been slow because there isn't a lot of data out there for these high tiered biodegradation endpoints, which I think has meant that there hasn't been a huge amount of decision making around persistence, which I think has led to ramping up the pressure to generate more data and to make more decisions around persistence. But I think that there's an unintended consequence here that we're going to make a lot of decisions based on perhaps some questionable data in the years ahead, given some of the shortcomings with these methods that are being used in the assessments overall.
Yeah, when I think about some of the other initiatives that are ongoing, which are great ideas like this is safe and sustainable by design, some of those other
Mhm.
regulatory initiatives to make sure that we are making the best decisions we can going forward, having learned from the past. It will be difficult to make those decisions well if we don't have an underpinning good data set. If really we cannot make comparisons between chemicals, either because we've not been conservative enough or because we've been too conservative, you're not going to be able to differentiate between things,
Mhm.
between the chemicals when you want to make a choice as to, is this truly biodegradable or more biodegradable versus this other option?
Yes. And if the tools that we're using are not sufficiently robust as well, then we won't be able to differentiate with precision between those chemicals that are really problematic versus those that perhaps, weren't, the microorganisms were having a bad day in the specific test that was run, when you spent 100 to 500,000 euros running that test on that one substance. So it's not trivial, I think, the challenge that assessing persistence causes. And I think as well, I mentioned earlier that it's not only about the testing itself. There are other examples of work that's been done to improve these testing methods, which Concawe has been leading on, like addressing substances that have poor water solubility and volatility, which in combination present huge challenges for some of these, for running and interpreting some of these data. But then the other thing is that in order to conclude a persist or conduct a persistence assessment, you have to look at all the data in its entirety and perform what's called a weight of evidence to actually assess the reliability and relevance of each individual piece of evidence and somehow combine that into a conclusion. And there's a lot of guidance in regulatory text around how to do that, but there were also a number of gaps when the rubber really hit the road of how that data should be combined together. And that, I think, is what led to yourself and also Ameliot from the cosmetics side to fund this persistence assessment tool that's been developed. So is there anything more you'd like to say about that?
Yeah, so it's funny because I was earlier this week going back and looking at what is written about weight of evidence. And when you look at, the text of how you're supposed to pull the weight of evidence together, it's very strong on where you're supposed to pull the information from and how you could grade it on reliability or on relevance. It's relatively strong there. But what decision you make at the end is, so you've pulled all this information together, you've got all of it. Now what? And um, there is so much room for subjectivity there. And I think this was part of what we wanted to help with the PAT was not to remove the sort of expert opinion, not to remove that subjectivity, but to make it transparent. And so when you know exactly how you have weighted the different pieces of evidence, you can have a better discussion at the end about whether the conclusion you've reached is well supported or not. If I just say, okay, I've got all these pieces of evidence, but you have bits of conflicting information, you don't know why, as the regulator or as the industry person, the registrant, you don't know why that decision has exactly been made. You can get an explanation, but the transparency, I think, is where the PAT really helps shine.
Yeah, I was really pleased to find that it did ultimately find its way into the new guidance that's been issued for the new hazard classes under the classification, labeling, and packaging regulation. It wasn't clear that it would be included in that guidance, but I think the fact that it is signals that the value of the PAT is recognised in terms of how it can help support some of these very complex assessments.
Yeah, and I think having it mentioned there will help build familiarity with it and hopefully demonstrate sort of the value of being able to assign concrete weights with some transparency and justifications. I know that there was some trepidation because the tool hadn't been out very long. It didn't have a big community that was already experienced with it, but it's a chicken and egg issue there. I think if nobody knows about it, nobody can use it to build that familiarity. So, yeah, I am also hoping that it gets used, that its benefits are made apparent, and that it gets greater uptake, basically. Uh,
Yeah,
yeah.
yeah, fingers crossed. So you mentioned about some of the work that Concawe has done in summary, and just to kind of try to recap that, you mentioned some models. So I think that the two of the important modeling tools that Concawe developed were the PETROTOX and PETRORISK tools. One is for assessing aquatic ecotoxicity. One is for actually performing the risk assessment, which integrates ecotox fate and use patterns to actually assess risks for specific uses. Both of those tools have been out for a number of years, and there have been various discussions with the regulators over the years. And I think that there's general reasonable acceptance of the tools and the confidence we can have in them, and that they're both based upon a concept called hydrocarbon blocking, which I think you sort of touched on in terms of how those hydrocarbon blocks are informed. And then I think another area that is really, another one of the challenges is around what's called the PBT assessment:
Hmm.
persistence, bioaccumulation, and toxicity, which, we touched a little bit on the P and the B already. And more recently now we have PMT, where M stands for mobility rather than bioaccumulation. Is it anything you'd like to do to sort of discuss around those aspects?
Yeah. So, with regards to the hydrocarbon blocks, right? I think they work really well for PETROTOX and PETRORISK. So the idea of those blocks there is that we're grouping together uh, constituents, or assuming that there are groups of constituents that, based on their volatility and their carbon number, are going to uh, come out together when run on an instrument, on an analytical instrument, the two-dimensional gas chromatography, just to be completely technical. So
Mhm.
you can have information about the composition of your substance by saying, okay, I think this group of peaks in one area represent, a certain family of chemicals. And then you could say, okay, and I have about this percentage of that family in my whole substance. So that way you can break down your whole substance composition into percentages of different families with different carbon numbers. That allows us to make predictions about the toxicity, but also the fate of those particular constituents. And then we can add that up together at the end for PETROTOX or PETRORISK. When we try to apply that same blocking structure for PBT, it gets a little bit more complicated. So for toxicity, we're using what's called the target lipid model. So this is like an underpinning model
Mm.
uh, with a predefined mechanism of action that then allows you to predict toxicity. And toxicity is what's driving most of the work in PetroRisk and obviously in PetroTox. When we're talking PBT, we're talking three different properties: persistence, bioaccumulation, and toxicity. There are going to be different reasons why chemicals behave the same from a persistence perspective, which is different from a bioaccumulation perspective, which is different from a toxicity perspective. So if you're trying to group chemicals together that have the same properties for P, B, and T, you might be looking at like a Venn diagram of chemical
Mm.
families. And so for grouping constituents together for PBT might have to be different than how we have historically done it for the hydrocarbon block model.
Mm. Mm. Yeah, and it's a hugely vexing challenge, right? It's what we call a wicked problem. One of those problems that are so difficult, you need really creative ways to solve them because, as you said, you need to look at not only individual endpoints of persistence and that bioaccumulation, but you actually need to look at them together. So that makes selecting one or a limited number of individual constituents to really focus in on more problematic because one might be clearly a worst case for persistence, but it might not be of any relevance for bioaccumulation or vice versa. So you naturally need to try to cover a wide chemical space with your assessment, but you don't have the resources to go to the degree that you might assess a monoconstituent substance, a single substance
Yeah.
under a normal process. And I think the other thing to recognise is that this is a requirement for all substances, all organic substances registered at greater than 10 tons per annum. They all need to undergo this PBT assessment. So the fact that it's so difficult for petroleum substances and Concawe with all of its resources, how can we expect this to be implemented across the board for the 30% of complex substances that are registered? And it seems that there's a bit of a willful denial, at the moment in terms of what's achievable under reach for complex substances in this area.
Yeah, because also PBT is supposed to be on a constituent basis, right? So it's not supposed
Yeah.
to be for the whole substance, it's for all the constituents in that whole substance. That is why we try to group them together using the hydrocarbon block approach, because that simplified thousands of constituents into, 400 blocks or so. And while 400 is still a lot, it is it's simpler than thousands and or millions. Yeah.
hmm hmm Yeah, and it's probably worth saying here as well that with the introduction of PMT, persistent mobile and toxic, as a concept, that this has created a whole extra layer of challenge and complexity to these assessments, because in the past you could use an either or approach to say, if something's clearly not B, then we won't worry about it. But now that we also have to look at M, many of the things that are not B are very likely to be M. The ability to kind of filter out some parts of a UVCB composition that were clearly not bioaccumulative and focus your efforts elsewhere seems to have been lost, because you now need to look to see whether those areas are PMT or VPVM. So you don't even need to look at toxicity. You just need persistence and mobility, or persistence and bioaccumulation. So it makes the task, an order of magnitude more challenging.
Yeah, and then exponentially more because you also have to look at all the metabolites, the degradation products,
Yeah.
um, for PBT and PMT. And I mean, not to complain. I don't want this to sound like a complaint. It's a lot of work. It is necessary because you do, right? Why are we doing all of this? Because you don't want to end up putting something out in the environment that's going to stick around and be a bad actor. But at the same time, how can you do all of these assessments? Yeah, there's a lot of screening approaches that are necessary. We need to be able to screen things out so that we can prioritize, we can focus on what's important. And yeah, I think that ability to focus gets really difficult when it's like, so many constituents.
Yeah, I think I have to say again, my concern with all of this is that as regulatory processes continue to evolve and we add new things in with limited evidence up front as to what their their ultimate implications are, it feels to me like we might be setting up a lot of things in train that we don't have a good foresight of where we'll end up in the end in terms of the
Mm-hmm.
chemicals that will ultimately be found to be fulfilling these criteria. And so, whilst a lot of the ambition to kind of improve and make, be more protective of the environment is, perhaps laudable, I think that we may find the outcome to be, let's say, less than desirable in terms of which chemicals ultimately are found to fulfill these criteria, and also the costs that are involved in getting there as well, when you take everything into account in totality.
Yeah, and I guess there's this interplay or push against, what are two things pushing against each other?
Mm,
There's this tension
yeah,
between conservatism and trying to make sure that you're making conservative decisions to protect people and the environment, and the ability to prioritize things.
yeah,
Because all of a sudden you've ramped up the number of chemicals that you think are of possible concern, and you will not be able to see the trees for the forest because you've got so
yeah,
many trees. Yeah.
yeah. If everything's a priority, then nothing is a priority. Well, Del, I'm
On that
thinking
chipper
that
note,
our conversation has gone in many different directions, and there was more that I wanted to talk with you about, and there's more that we could probably go on for the same amount of time again. But I think this is a good place to wrap things up. So I just want to say thank you so much for taking the time to talk with me today. I've really enjoyed our conversation. I knew it would be really interesting, and I hope the audience has also found it interesting. I ask all of my guests a couple of quick fire questions just at the end, just to wrap
all
things
right.
up. So, yeah, first one is, what is a big goal, and this could be personal, professional, that you're working towards in the coming months?
Oh, a big goal. There are a number of papers that I would like to have some papers finished. That's a big goal that I'm working
That's a
towards.
goal that we all
I'm
share.
winking at you, Chris.
Message received. Thank you. And the second one is, if you had one piece of advice for either yourself or somebody else starting out in their career today, what would it be?
Ooh, something that has been really helpful but difficult to do is when you are at workshops or when you are attending conferences to go up and talk to other people that you don't know. It's really tempting just to stay with your friends, but You can read all the papers in the world, but having a conversation with somebody who is an expert in that field or who can give you the cliff notes is really beneficial. So I would say go talk to people, even if it's hard, especially if it's hard.
Very, very good advice. Thank you, Del. Thanks, everybody who's been listening. Your time is really precious, and I'm really grateful you've decided to spend it with us. If you've enjoyed the podcast, please follow it, like, subscribe, comment, and spread the word about the Chemical Journeys podcast. We've got more conversations like this to come, so I look forward to catching you next time. Thanks very much, Del.
Thanks, Chris. Thanks for having me.